Dr. Biteshwari, you are there? Hello, Dr. Biteshwari?
Yes, ma'am, I'm here.
Oh, okay. So, Sudha madam, we can start?
Yeah, you can really.
Okay. So, I will request Dr. Biteshwari to start the proceeding.
Honorable President, Dr. Sudha Tiwari of National Sakti, Resource Person, Professor K. Solochana Devi, Microbiology Department, Institute of Medical Science, Professor Atal Gupta of Manipur Sakti, faculty members of Manipur University, faculty members of different colleges, administrators, or members of Manipur Sakti, and participants. Good evening to one and all. Today, Manipur Sakti in collaboration with Manipur is celebrating Science Week. I, Dr. Biteshwari Khangembam, Faculty of Environmental Science, Tamenglong College, on behalf of Manipur Sakti, will say a few words. I welcome Dr. Sudha Tiwari, President of National Sakti. We are lucky to have with us who is a scientist, botanist, innovated 15 products and known for developing bioplastic, which is biodegradable. Acting burn. So, Madam, we are thankful for your kind gesture to be present here in this webinar and given your consent to preside over today's webinar. I also welcome our resource person, Professor K. Solochana Devi, Microbiology Department, Regional Institute for delivering lecture on the topic Women Scientists at the Forefront of Fight Against COVID-19. I welcome Professor Gupta of Manipur Sakti, who is inspiring us to have a program in Science Week. I also welcome all the participants present here. Women scientists across the globe have been at the forefront of fight against the global pandemic COVID-19. Women play a very important role treating patients and assessing the pandemic devastating economic and social impact. Women have been extending enormous contribution to the global effort to tackle COVID-19, leading research into the virus, creating trackers and developing a vaccine. So this webinar is organized to discuss about women scientists at the forefront of fight against COVID-19. Now I request Dr. Niru, Assistant Professor of Zoology Department, Moirang College, to introduce our resource person. Professor K. Solochana Devi, Microbiology Department, Regional Institute of Medical Science, to the participant. Dr. Niru, please.
Very good evening to all of you, respected resource persons and all my dear friends. Let me first of all introduce today's resource person, Professor K. Solochana Devi. She is a professor by designation in Microbiology Department in Regional Institute of Medical Science. Her date of birth is 1st June 1956. And educational qualifications, she has completed her MBBS in the year 1976 at RMC, now then finished her PhD and Evel Gold bronze medal in PGI-MER, Chandigarh Medical Microbiology in the year 1985. Research and training experience wide. She has completed her senior resident in the year 1985 in Chandigarh in PGI-MER, and work is completed in the of Vero. She is a WHO fellowship from October to November 2011 at Bangkok Hospital and work on STDs and AIDS. She has done her research specialization in antimicrobial resistance and in viral hepatitis. She has published around 70 publications both in national and international level, and completed a project on networking of gonorrhea surveillance in India under ICMR. And at present, she's working on projects of viral research and diagnostic laboratory under DHR ICMR. And she is also doing project on surveillance of drug resistance infections in Manipur under ICMR, and also capacity building and strengthening of hospital infection control to detect and prevent antimicrobial resistance in India under CDC, ICMR, and in AMS. She is also a member of scientific bodies like life member of IAMM, SIHM, IMA, HISA. It's very high fellow, though I don't know all those short names. And then she is also an examiner of different universities like WBUHS, SSUHS, NEHU, Tripura University, AIMS, and New Delhi, etc. We're fortunate to have you, ma'am. Such a great fellow in our meeting.
Yeah, women fight at the forefront of fight against COVID-19.
Good afternoon, everybody. Dr. Asha Gupta ji, Dr. Neeru ji, and the other participants. I'll be talking on pandemic COVID-19 and overview and women scientists in the fight. So, 6th International Day of Women and Girls in Science. It was on 11th of February 2021. The theme is very appropriate. Women scientists at the forefront of fight against COVID-19. First, before going to the scientists, I'll just tell something about the COVID-19 also. And this COVID-19 pandemic has demonstrated the critical role of women researchers in different stages of the fight against COVID-19. Development is a disease, and the causative agent is severe acute respiratory syndrome coronavirus 2. That means, in short, SARS-CoV-2 among the group coronaviruses. What are the symptoms? It vary greatly, ranging from none to severe life-threatening illness. Why it is important to discuss about this COVID-19? It is a new virus, new strain, previously not identified in humans, and transmission patterns, severity, clinical features, risk factors for infection remains limited. Being highly transmissible, this novel coronavirus disease, also known as coronavirus disease. So the long term of COVID-19 is the coronavirus disease 2019. This has spread rapidly all over the world. The characteristic of this is it has the ability to infect multiple hosts like the bats, the camel, the pangolins, the human beings, etc. And there is high frequency mutation. So mutation is very common in RNA viruses. So this, like this COVID, SARS-CoV-2, CoV-2 is also very has got very high frequency of mutation. Instability of RNA-dependent RNA recombination nucleic acid tests remain the gold standard for detecting the presence of the virus. So it has spread, as I've taught before, it's spread all over the world, except it's very less in Greenland, and all over it is spread, this global pandemic. Let us see the timeline of the key events of COVID-19 outbreak. It was in 8 December 2019 that the first case was recorded. But Chinese government, they have reported cluster of pneumonia of unknown cause. So this is the alarming situation without any known cause. There's 27 cases of pneumonia. So then in January 9, China announced identification of this novel coronavirus as a causative agent, and so on. It has been spread from many provinces across China. Then 30th of January 2020, WHO declared it as public health emergency of international concern. This is from there it has become worldwide public health emergency. WHO named this virus is SARS-CoV-2, and WHO named the disease COVID-19. So this is continuously spreading, and 11 March 2020, WHO defined COVID-19 as a pandemic. So then till today, till 16th of February, this many cases have occurred worldwide, and this many deaths. Okay. So let us see the virology of this SARS-CoV-2. This virus is a positive single-stranded RNA having 29,903 bases, and it is an enveloped virus, and it has got four structural proteins: spike glycoprotein on the surface, and a membrane protein, nucleoprotein, genomic, then the envelope protein E. There is genetic similarity of SARS-CoV virus. That means which occurred epidemic in 2003, 79%, 79.5%, and with coronavirus, it is 96%. The receptor for this virus is angiotensin converting enzyme 2. Let us see all coronaviruses which can infect human beings. This causes mild, self-limiting, just like common cold. is occurred in China. And another is in 2012, Middle East respiratory syndrome coronavirus that occurred in Saudi Arabia, and it was from camel, in contact with camels, infected camels. So these coronaviruses are, they can infect multiple hosts. So it causes severe respiratory infections, very fertility rate, with increased fertility rate. Then the last one is the coronavirus 2, SARS coronavirus 2, which occurred in 2019 and 20, and originated from infected seafood in Wuhan, in China, and also bats at market, in a Hunan market, seafood market in Wuhan. So it has increased and it has spread far and wide, and it becomes a public health emergency of international concern. There are clinical cases, deaths, and asymptomatic cases, transmission to different parts of the globe. Okay. So let us see the life cycle, how this coronavirus, SARS coronavirus, infects a cell and replicates. This is the coronavirus, SARS coronavirus 2, SARS-CoV-2, and this is the human cell, and this is a receptor, SE2 receptor. So it is attached to this through this receptor, it is a polymerase, that is RNA dependent RNA polymerase, and which leads to polymerization of this replication of RNAs. And it also transcribes the subgenomic proteins. First, it was by polyprotein. It is by the protease. It is broken down into different. This is a spike, membrane, envelope, etc., with the endoplasmic reticulum membrane. Then it is the virus then assembles with the nucleic acid and the polyproteins. Then it bursts out, comes out, and taking a part of the host cells as envelope. This lipid as enveloped. Okay. And one thing is that these ACE2 receptors are also present in different cells, macrophages, epithelial cells, microglia, neuron cells. That's why this virus can infect many cells and many organs. Pathophysiology, see, the virus goes to inside a human being through respiratory droplet, larger cases, blood oxygenated and deoxygenated blood. And what happens is that this SARS-CoV-2 infects the macrophages with the ACE2, and it is activated, producing many cytokines, interleukin 6, interleukin 8, interleukin 10, and tumor necrosis factor alpha, which attracts or all other immune cells, neutrophils, and other immune active cells. And because of all these things, there is heightened immune response. Cytokine storm is there. And there is also increased vascularity, increased porosity of this, and there is damage of the cells, pneumocytes of these alveoli and cell wall, endothelium, pneumocytes, etc. And all this because of this chemokine, inflammatory chemokines. It's not only in COVID-19, the pathophysiology is not only the virus attacking, damaging. It is also by the immune response, human immune response, cytokine storm, which is giving a very inflammatory response. So this can in fact cause acute respiratory distress syndrome. Other cardiac and respiratory can even cause Kawasaki disease. Whether COVID-19 it is Kawasaki-like symptoms are seen in COVID-19 patients. So the relation between COVID-19 and Kawasaki disease is under investigation. So other clinical manifestations, as I have told before, it can any system can be involved, organ involve lung, respiratory, pulmonary, brain, neurological, gastrointestinal, heart and cardiac, kidney, renal, like this. Because the receptor SE2 is present all these things, and this can be spread through respiratory route, through the olfactory nerve in the nose to the brain, or it may be through blood circulation also. So let us see the women scientists working on pathogenesis. Who are working on the pathogenesis? Ben Zhang. So she herself is a pediatrician in China and wanted to understand why some children are susceptible to infections than others. So she performed postdoctoral work at the National Institute of Allergy and Infectious Diseases. Afterward, Zhang became a postdoctoral fellow at the Rockefeller University in the laboratory Jean Laurent Casanova, MD, PhD. So she hopes that the pandemic-related research will allow her group to better understand Kawasaki disease. This is Kawasaki disease, a serious disorder that has been associated with COVID-19 in children. Akiko Iwasaki is professor in immunology, molecular, cellular, and developmental biology of the Yale School of Medicine. She's an investigator in Howard Hughes Medical Institute. Her study was to understand the immune responses to COVID-19 patients, the role of interferon in this disease. Necessary protective but causing harmful effects. Then she also tries to understand immune response. What are the different immune response? Whether it is protective immune response, whether it is the not protective but lead to disease. So she's trying to work on the disease trajectories that reflect patient-specific aspects of the immune response, cytokine or antibody production, T-cell response, viral load, etc. Because it may be different from individual to individual, and she wanted to see the patient-specific aspects so that the clinicians can tackle individual patient-specific treatment protocol, and the cytokines, all the markers can be developed. Then another study is the sex differences in SARS-CoV infection, why males are reportedly more susceptible to COVID-19. Her team described how they investigated sex differences in viral loads, antibody titers, cytokines. So continuity was important because they had just started uploading sequences of SARS-CoV-2 onto online genomics engine NextTrend, in collaboration with 2014 to track flu viruses diversity and help predict the next flu strain. So as we know, flu strains also always changing, mutation and the new changes. That's why we have to prepare vaccines for each and every year. The roughly 40,000 cases currently in the system is a drop in the bucket compared to the number of COVID-19 cases. What? There is much more likelihood that we haven't sampled someone than we have. Unquote. So COVID-19 sees also against the second wave of this COVID-19. She believes that COVID-19, the virus, is empowered by our behavior and our actions, how we respond. So it all depends on how we respond. The SARS coronavirus spreads in the future. Rachel Graham uses large sequence sets to study how the virus transcription and program contributes to replication and virulence. So as we know, this subgenome proteins, SMNE proteins, the transcription is this assistant professor in epidemiology, and she's working on the host virus interaction, and she's using animal models such as humanized ACE2 transgenic mouse. So she's studying on this, the receptor and the virus, how it attacks and everything. She's working on this. Viviana Shimon, she has analyzed the genetic diversity of SARS-CoV-2 circulation in New York City and how the virus was introduced. The first COVID-19 case was diagnosed at Mount Sinai in February 29. Only then could the Simon team grow the virus and sequence it. They are also working on assessing the durability of antibodies and determining the degree to which antibodies are protective. This is very important work she's doing. So let us see viral setting and a period of infectivity. The incubation period is about 2 to 14 days with a median of 5 days. This SARS-CoV can be transmitted prior to development of symptoms. So the patient pre-symptomatic stage, it can transmit disease to another person. Another point we have to remember is that Why by RT-PCR for more than 14 days, up to 20 days, 30 days like this? There is difference. Some day the RNA cleared in 1 week or maximum in 10 days or 14 days. But the presence of this viral RNA doesn't mean that the person is infectious, because this is RNA, not a virus, complete virus. And it has been found that when the RNA is present in the samples after 10 days or 14 days, the virus could not be isolated. So they are not live viruses, only the RNAs. Transmission is through close contact, range of 3 to 6 feet, via respiratory droplets. The person, when he coughs, sneezes, or talks loudly, then the virus comes out droplets. Droplets, that means about 5 microns in size. And these droplets can remain suspended in the air for about a short time. And within this range, if another person comes in contact with this person, infected person, more so in a closed chamber or closed room, he is very likely to be infected. But this virus can stay viable on metal, glass, plastic surfaces for up to 72 hours, or on cartons for about 24 hours. So these viruses may be suspended on these surfaces, or maybe on the table, on the laptop, on any surface. Then what happens when we touch the surface and touch our nose or eyes or mouth? Then there is chance of infection by this virus. Another mortal infection is fecal-oral. That means touching or smelling of fecal matters can also get infected. Can lead to infection to another person. And fecal microbiome transfusion, which is used for treatment of pseudomembranous colitis, severe form of pseudomembranous colitis, that also can transmit the disease. The virus stays also. Again, this can be transmitted from time in an laboratories, but some super spreaders are also found which infects more than 10 persons. So to summarize again how this infection occurs, the keep touching and all these things. Age as a major risk factor, we know older than 60 years, severity is more. So the mild cases will be in less than 50 years. It is like this. Okay. Laboratory diagnosis. So first of all, we have to take the samples: oral, oropharyngeal, nasopharyngeal, or nasal swab. For this, we have to use precautions with personal protective equipments, and there's with the hands introduced inside, and it is taken like this, doctor standing outside. So real-time PCR is the gold standard. Other modalities are TrueNet, cartridge sprays, Curtis base nucleic acid amplification tests, LAMP, and the serology, antigen detection, rapid point of care tests, antibody detection, etc. Let us see who are the women scientists behind India's the testing kits. We have heard of this Minal Dakwe Bosle, Sepali Desai, Mitali Patel. They developed India's first COVID diagnostic kit, my lab, and it's very interesting story about Annie L. Wiley drew the attention that she had been using detect Streptococcus pneumoniae from saliva samples of asymptomatic carriers. So based on this, she also worked on detection of antibodies in antibodies of SARS-CoV-2 using saliva to assess past SARS-CoV-2 exposures. Very interesting study. Let us see disease management. There is not a foolproof treatment for this. We have to support care, supplemental oxygen, maybe ventilator, maybe required, and antiviral agents, remdesivir, favipiravir, lopinavir, etc. Hydroxychloroquine, corticosteroid, dexamethasone, azithromycin, etc. And whenever there is complications or coagulopathy, we have to use anticoagulants, heparin, and others. Role of BRDL in containment of this COVID-19. So, viral research diagnostic laboratory. There are about 106 functioning VRDLs, 9 regional, 22 state level, and 74 medical college level in India. What are the main objectives of VRDL? This includes creating infrastructures for timely identification of viruses and other agents causing morbidity significant at public health level, and specifically agents causing epidemics or potential agents for bioterrorism. So it is right time that VRDLs were established so many all over India, and we could diagnose this COVID-19 during a short period of time. Developing capacity for identification of novel and she is instrumental in setting up all the virus laboratories. This is just after the outbreak, pandemic of influenza 2009, H1N1 2009. After that, the viral research and diagnostic laboratories were established. She is also one of the scientists who investigated the viral outbreaks such as enteroviruses, arboviruses, dengue, chikungunya, JE, Zika, etc. So she is also responsible for developing management guidelines and also acute encephalitis syndrome in different parts of India. So we have under this ICMR, Department of Health Research, and the ICMR, this viral research diagnostic centers are there. Priya Abraham is the director of ICMR, National Institute of Virology, Pune, and the first testing of COVID-19 was done in NIB Pune only. We used to send samples for confirmation of our diagnosis, lab diagnosis before we start doing ourselves. So with this, the RT-PCR, it was taking about 12 to 14 hours for one test. Now it has reduced to about 4 hours. It is achievement of this NIB Pune. Renu Sharup again is the working the ministry administrator and the key personnel in formulation of biotechnology division in 201, and also she is very much involved in research to develop coronavirus vaccine. Billa Rajesh, another health administrative IS officer, health secretary who is very much interested in the coronavirus, fighting against coronavirus. So this is state level VRDL RIMS Impal, and we have under me we have 7 staffs, 2 technicians, 3 scientists, and 2 research assistants, and these 3 are all ladies. So as soon as we heard that WHO, it also shows how to don and doff personal protective equipments, how you wear all these things. This step by step is also very important to know donning and doffing. Donning is somewhat okay, but doffing is very important because we have used PPEs. We have to take it out in such a way that it cannot infect another person, and disposal is another issue. So in VRDL, so far we have done this much by RT-PCR, and this much is the positive. And this is from April to June. July, we started testing in last week of March, and we got the first positive case in 1st of April. Then onwards, we have these positivities. And you know, the samples are July, September, it was peaked, and it's reducing, and it is coming down. But if you see the trend, the samples are coming down, but we had a very tough time during this time. Well, state government has decided to bring all the people staying outside Manipur by train, by bus, by aeroplane. We have to test all of them. And per day, we used to test even 1,000, more than 1,000 also. And we didn't have any time to even our scientists and our staff stayed up to 11 p.m. in the night, starting from 6, 7 a.m. in the morning. This is the age and sex differences of the patients. And his peak is in the 20 to 39 years old. This is the maximum number of patients, positive patients. And if you see here, compared to the female and male are much more infected. Another research work we are doing is the molecular epidemiological analysis. We are also participating in whole genome sequencing of SARS-CoV-2 of different parts of India between January 20 to August 20 in NIB. Of course, we have started from March end. So in India, they have started from this thing. So the study is completed, genome sequencing completed. The inclusion criteria for samples are that appropriate storage of samples at minus 80 degree, cycle threshold less than 30, and samples are packed in dry ice in triple layer packaging as per guidelines of International Air Transport Association. And these are transferred to nodal lab, that is ICMR NIB Pune, for sequencing and analysis. How we prevent? Though fairly obvious, the best way to prevent infection is to avoid being exposed to the disease. So,
Hello, ma'am. Is this chemistry class?
yes.
Oh, sorry for this.
Uh, uh, Everyday preventive actions to tackle the spread are always recommended, like washing hands with soap and water, avoiding touching ears and all these things, dispensing frequently touch objects and surfaces, wearing face masks for healthcare workers, droplet and contact precautions, and use of PPE, including N95. So next comes the vaccines, the most important for prevention of this COVID-19. There are four types of vaccines: RNA-based, mRNA, that is Pfizer, BioNTech, and Moderna. There are two candidate vaccines, and another is the viral vector-based vaccines, Oxford, AstraZeneca, vaccines. Maker, Dutch virologist and professor of virology at the Medical Center of the University of Amsterdam, global head of viral vaccine discovery and translation in medicine at Johnson and Johnson's Vaccines and Prevention. Elena Smolly, it is the Russian Sputnik virus vaccine. So these are scientists to conclude whatever research we are doing, whatever findings we have, this is just tip of iceberg. Animal origin, cross-species infection root of SARS-CoV-2 yet to be uncovered. Virus-host interactions remain largely unclear. Here is very important. This is the key to break the chain of transmission. This is the responsibility of each and every individual. So let us see again what practice social distancing. And I just want to show the how do's and don'ts of wearing a mask. I have seen many people wearing masks like this, like this, like this. The correct way is this one. Thank you and stay safe.
Thank you. There is no queries, then I will move on. Now I request Dr. Niru to introduce President of National Sakti, Dr. Sudha Tiwarizi, to the participants.
Okay, let me take this opportunity to introduce Dr. Sudha
Niru,
Tiwari.
just
Hello,
one second.
Niru.
Just one second. Let me ask Madam one question, small question. Madam, you have talked very nicely, many aspects, and talked about personal protective behavior at the end. I just want to say that we know that this SARS COVID, as we are seeing, so many strains are coming up. In UK strain, we have seen African strain, we have seen, and we have also talked that many phenotypes may come up also.
much difference between the severity of disease by this mutated virus, but then it is found more transmissible, more stickier. The virus spike protein is more stickier to the SCE2 receptors. And about the immune response of this, even the for for SARS-CoV-2 also, the immunity, how long it is protective, the vaccine, after vaccine, how long it will be protective is not yet known. It is under process. But the strains. Okay, so every year we have to the circulating antigen, circulating virus strain. And after knowing the circulating viral strain in that year, in that period, then we prepare vaccine again. So one vaccine may not always protective to others. This is this information comes from the experience with the influenza viruses. They are also RNA viruses prone to mutate, prone to develop new strands, new antigenic variation, etc.
Okay, thank you, madam. Thank you for your answers. Now I will ask Neeru to continue. Dr. Neeru.
And who told him?
Neeru, we could not listen you. Neeru.
MGIRI, Mahatma Gandhi Institute of Rural Industrialization, Warda. And so far, she has innovated about 15 products, to name few, like transparent soaps, mosquito coil, hand wash, then herbal face pack, shampoo, which are daily use requirements, and like which are without any hazardous chemicals, and for field bio manures and soil testing kits, etc. And she has also motivated about 300 entrepreneurs throughout India, and working in a DIC project under Ministry of MHRD. And she has also, the best thing about her is she has developed bioplastic, which is, we all know it is a biodegradable things, and it is made up of like biopolymers like starch, and which is safe for both animals and humans as well. So thank you, ma'am, for being with us this evening.
Thank you very much.
Thank you, Dr. Neeru. Now I request Dr. Sudha Tiwari, President of National Sakti, to give a presidential speech.
Thank you very much. I won't take much time because already we have, I think, the time is over. Ji, nice and wonderful Science Week. And it's really commendable that in such a short span, because few days ago only Manipur Shakti team has come up, and in such a short time, arranging such a beautiful and very informative scientific lectures is a very commendable task, and I must congratulate Asha ji and her team for this particular uh, events. Now, I want to throw some light why Shakti thought that this type of Science Week should be celebrated, and what is the need of attracting the girls and the 50% of our population towards science. We see that if you see the percentage of the PhD holders, you will find that there is 30% of them are women. And when you see the workforce in science, you see a mere 17%. And if you go to the directorate post or the leadership in the scientific institutes, it remains only 2%, and it's a really a pathetic condition. And this, there are many reasons for this type of degradation as you go up. There are career breaks are there in women's life due to the family constraints are there, then marriages are there. So there are many reasons for this, and actually we have conducted a very good webinar on this particular what is the necessity and what are the hurdles and what are the constraints which are there in strengths is there, and we have this was held with respect to the preparation of science, technology, and innovation policy, in which the public opinion was taken, and we had taken a very good webinar on this particular thing, and we have recommended many few points for this particular policy, and we are really happy that when we saw the policy, we are happy to announce that most of our recommendations are incorporated into the policy. And there are this new policy has really many new things which will help a woman to come up in the science. Now, for this, Shakti is doing a lot of work how to attract girl into the science, because see we at a very good you can say that at a very good position don't see the real side of real picture, because still there are few states in India wherein the education for the girl and the coming up of her as a scientist or as taking up as a science as a career is really a thing which should be thought of. And actually, we Shakti had also conducted a very good survey recently on women scientists throughout India, and a finding which I thought one's inherent you can say that what is our work is there, our duty is there, and that we prefer the our work, that also somebody may argue her work actually a fact that a woman by nature is very sincere and dedicated, and she tries to cope up whatever might be lost due to this particular preference, by working hard, by doing extra time, and so on and so on. And this has to be considered. But in a society, when we see that, this dual responsibility is sometimes I having such a particular consideration. Although we, most of us are lucky because we are a gathering of very successful women and who has come up as a scientist, as an academician, and but if you see actually the very ground reality, you will see that these things are there which has to be considered, and therefore uh, arranging such a week and arranging such a scientific lectures and week to celebrate the science day is a must, because this gives inspiration to the girls to come up and take up science. And it because see, there is a fear in the very basic, if you see the base of a education, you will find that there is a fear, not amongst all, but there is a majority of them are there who fear taking up science as their career. And give the science as a career. Science, technology, and innovation. You know that these are the things which on which the progress of any country depends, and to which, and we all know that 50% of us are women, and so therefore, if we attract the coming generations to take up the science as a career and contribute in innovation, science, technology, and innovation, and making the India Atmanirbhar as we all see. So that is a must, and therefore this type of week are must. And I'm really happy to see the kind of the standards which this particular lecture series has. I have seen the flyer, and I have seen that this is a very good initiative which Asha ji and her team has taken, and those who students who are listening to, they must have got the inspiration, and they will be successful in their lives. And I won't take much time, and thank you very much for calling me, and I once again congratulate the team. Thank you very much.
Thank you, Dr. Sudha Tiwari. Now
Thank
I
you, Dr.
request Professor
Biteshwari.
Asa Gupta of Manipur
Honorable
Shakti to come
Sudha
forward with
ji,
vote of thanks.
National from National Shakti, our president, and also president for today's function. We have our resource person, Professor Solochana ji from RIMS, our respected resource person for today's program. We have the other Shaktis here, Dr. Irom Bilal, I can see, Dr. Rajashri Kashalkar, I can see. We have got Professor Ratna from Manipur University. We have Professor Jha from Mithila University. I'm really happy to see you all here in this meeting. We are observing this Science Week. Madam has already told the objectives of it and what we want, what Shakti want to achieve from it. Honorable Madam, I'm so much thankful to you for sparing your valuable time for us. We know that you are daily busy in morning and evening in different schedules, presiding some function and giving some addresses, etc. But still you have found time for us, and very humbly, I have just in morning have one telephone call to Madam, and Madam accepted immediately. So I'm really very much thankful to you. You are our role model, and we will always follow your footsteps. Thank you, Madam. will like to keep COVID-19. She talked about virology, the belonging to family coronaviridae. She talked about ACE2. She talked coronavirus has multiple hosts. She talked that it can affect many cells and organs. It continues to evolve. Then she has talked about clinical manifestation of this infection. Then she has talked about who are the women scientists in pathogenic field, what they are doing, like Jian Zheng, then Iwasaki, Ima, then Rachel, Graham, Lisa, Viviana, and their contributions. They are all in different directions. Then she has demonstrated the crucial role women is playing in advancing knowledge, in developing techniques of testing and creating vaccines also. Then she has told, the transmission ways, how it can be transmitted, the age as a major risk factor. She had talked about laboratory diagnosis. She has talked women scientists, Indian women scientists who are at the forefront. She has talked about Shifa Alida, she has talked about Mitali Patel, Minal Bhosle. Then she has talked about disease management. Then where she is working, she is having a great responsibility because it happens to be one of the 9 regional laboratories in the country, and she is heading one, that is VRDL laboratory. And then what is its role in containment? She is acknowledging Nivedita, Dr. Nivedita, Dr. Priya, Dr. Renu Swaroop from DBT, Krishnan Vaccine Laboratory. Then she has told saying that this is tip of iceberg. After saying so many things, and then still there should be ways and means and intensive study, it should be continued. And she is impressing us that there should be personal protective behavior. Madam, so much we are thankful to you for giving so nice information. And then really the whole Shakti team is acknowledging your efforts, your tremendous, you have spent time for preparing all these slides and giving this lecture. Now I'm also thankful to all the participants, our Shakti members, who are wholeheartedly participating in this function. And then again I reciprocate, again I acknowledge the help received from National Shakti, Sudha Tiwari madam, and our resource person, Professor Solochana ji, in making this function a great success. With permission of chair, I declare the meeting as closed, the program as over. Thank you so much. Thank you, Sudha ji. Thank you, Solochana ji.
Thank you, ma'am.
Thank you. Thank you.